Mangrove forest is a unique ecosystem with both terrestrial and aquatic characteristics that maintains the balance between the land and sea. Mangroves are shrubs and trees that thrive in the intertidal areas of tropical and subtropical shorelines. They grow within the coastal zones at the middle to upper intertidal zones, excluding the levels of the mudflat areas and the seagrass beds. The mangrove forest is a unique community of trees occurring in a very defined zonation pattern. The mangrove zonation is due to the type of substrate and the tolerance of the species to salinity and inundation. There are 3 zones of mangrove habitats: seaward, midward, and landward zones. Unlike terrestrial plants, mangroves live close to the shorelines and evolve with special structures that tolerate saltwater and oxygen-poor soil. These special adaptations maintain the integrity and diversity of mangrove habitats. Mangrove habitats provide ecosystem services such as fisheries, coastal protection, biofiltration, habitat to wildlife, and carbon sequestration. In the Philippines, about 45 to 50 mangrove species are recorded and rank 16th in the world in terms of mangrove cover, with 400,000 to 500,000 hectares recorded in 1920. However, only 300,000 hectares remain in 2015. The decimation of mangrove habitats are caused by the conversion to fish ponds and salt beds, the clear cutting of trees for firewood and other domestic uses, and the reclamation for industrial or other development purposes. To conserve and sustain this habitat, regular assessment and monitoring must be conducted to provide a science-based approach in identifying management interventions. The DENR Biodiversity Management Bureau provides guidelines in the assessment and monitoring of our coastal and marine habitat. Follow the belt transect method when sampling the mangrove zones in order to determine the species, diversity, and their conditions. Prepare the following materials: 100-meter transect tape, GPS device, camera, data sheets, pencil, field guides, diameter tape, Haga altimeter, graduated pole. When establishing the belt transect method, identify the high water mark, then lay the baseline transect parallel to the shore. Follow the contour of the shoreline. Lay the transect lines perpendicular to the baseline transect at every 100-meter interval from seaward to the most landward zone of the mangrove forest. Establish a nested 10-meter by 10-meter plot alternately along the transect lines, with an interval distance of 25 to 50 meters for patches or strips of mangroves and 100 meters for contiguous. Record all trees with greater than or equal to 5 cm diameter within the 10-meter by 10-meter quadrant. Then create a 2-meter by 2-meter plot inside the 10-meter by 10-meter. Take note of the small trees with less than 5 cm diameter. Label each quadrant during photo documentation. Provide codes for the site, transect, and quadrant. Record the coordinates at the start and end of the transect line. Within the 10-meter by 10-meter plot, identify the trees with greater than or equal to 5 cm diameter. Measure the following: the diameter at breast height DBH with a calibrated diameter tape and/or tree caliper. Measure 1.3 meters from the ground up to the trunk of the tree to get its diameter. Measuring depends on the tree forms. The total height, TH, measure from the base of a tree up to the tip of the crown. The merchantable height, MH, measure it from the base up to the first major branch of the tree. The crown diameter, CD, measure the widest length, W1, of the tree canopy through its center and the canopy width to the widest length, W2. This determines the condition of the crown cover. Identify and count small trees, seedlings, and saplings with less than 5 cm diameter within the established 2-meter by 2-meter plot. Record and identify correctly all associated flora such as shrubs, herbs, vines, and ferns found in each quadrant. Record and photograph per quadrant the flowering and fruiting season and the presence of threats such as cutting, debarking, presence of barnacles, and land use activities. Document all animal species such as avifauna, mammals, arthropods, herpetofauna, and invertebrates. For assessment of mangroves in riverine areas, lay the baseline transect from edge to edge of the riverbank. Establish transect lines perpendicular to the baseline transect. Encode all data in a Microsoft Excel worksheet. Compute for the importance value, IV, by getting the sum of the relative density, relative dominance, and relative frequency. This will determine the rank relationships of species in the ecosystem. Density is the number of individuals per species occurring per unit area. This helps determine the health status of a forest. Relative density is obtained by dividing the density of the species by the sum of the densities of all the species sampled, then multiplying by 100. Dominance is the basal area, BA, or volume, V, of a tree. This determines the contribution of a particular species to the ecosystem functions. Compute the relative dominance by dividing the basal area or volume of species by the total of the basal area or volume of all the species sampled. Then multiply it by 100. Frequency is the number of times a plant species is present in the quadrats or sample points. This helps assess the vegetation trend. To compute for the relative frequency, the frequency of species is divided by the frequencies of all species, then multiplied by 100. Species diversity is a function of species richness, the number of species in a given locality, and species evenness, the degree to which the relative abundances are similar. Diversity index for the total area assessed is generated by using available biodiversity software. The estimated biodiversity value helps select the monitoring site. Biodiversity value, BV, evaluates the number of species recorded and the number of endemic and threatened species observed per transect. The range of the values obtained are used for a 3-point scale to rank the sampling unit. The average of the derived values are taken across all transects. To determine the endemism of the mangrove species, refer to other published literature and available online databases such as COS Digital Flora of the Philippines, the DENR Administrative Order 2017-11, and the IUCN Red List provides the list of threatened mangrove species in the country. In the absence of one of the criteria, the condition of the mangrove area must be assessed. Determine the value of the percent crown cover, regeneration per square meter, and average height. Use the formula to derive the information required for the analysis. For each transect, the condition of the mangroves shall be analyzed and classified per parameter, such as crown cover, average height, and regeneration, in accordance with the criteria provided in the Participatory Coastal Resource Assessment (PCRA) table for mangroves. Factors such as substrates and accessibility of the area are also considered in selecting a permanent monitoring site. In the PBMA, establish at least 2 hectares in size or more, depending on the size of the transect line. Divide into grids with a dimension of 10 by 10 meter. For easy location, place monument markers in every corner and center of each grid, or establish witness corners with a grid reception in GPS of at least 3-meter level accuracy. Diameter breast height, total height, distance and bearing from each tree from the center of each plot. Tag each tree for proper coding. Monitor the plant growth every 5 years after the baseline survey. Mangrove forest teeming with life is key to a healthy and balanced coastal ecosystem.